Okay, so today we're going to do a little bit more on kidneys for the first little bit, and then we'll take a break, and then we'll do the Kahoot review for Friday. So we're going to talk about sodium first, and then we'll talk about water, how water changes through ADH, as well as other effects with water regulation on the kidneys. Then we'll start getting into tomorrow. We'll deal with all the other electrolytes, magnesium, potassium, calcium, phosphate, all that fun stuff. So the daily filtered load of sodium is much greater than the requirement for sodium excretion. We want to keep sodium within tight regulation as well. Filtration and absorption are the only processes that affect sodium and water excretion. If we have to reduce extracellular fluid volume, either we have edema or hypertension, we give diuretics. Let me just change this. I don't know why it's doing that. Okay. So when you give a diuretic, the goal is to increase urinary salt and water excretion. All we're doing is we're shutting off the transport mechanism for sodium to get back into the cell. And if sodium and the chloride, the salt, is staying in the urine, then the water stays in the urine as well. So remember, wherever sodium goes, water is going to go along with it, usually to keep osmotic pressure gradients for the osmosis to be kept constant. So most diuretics are going to inhibit our tubular sodium reabsorption. So where is sodium mainly reabsorbed? Proximal tubule, 65 to 75% of it comes out there, reabsorbed there. Remember, we dump all of it into the Bowman's space, in the Bowman's capsule. That's our filtered load. We just dump it all in there, but obviously we don't want it to stay in there. We need to pull some of it back out. The bulk of it happens in the proximal tubule. Then in the thick ascending loop. Okay, that's important to remember. Thick ascending loop. Because when we start talking about different diuretics, they have different effects. They act at different parts of the nephron to alter your sodium reabsorption. And then fine-tuning distal convoluted tubule and the collecting duct. Remember, these are under hormonal control. Okay, when you get towards the end, it's all under hormonal control to fine-tune how much sodium we want to get back into the blood. So, of all the sodium that gets dumped into the Bowman's capsule and Bowman's space into the ultrafiltrate, only 5% of it winds up in the urine. We will reabsorb 95% of the sodium that gets dumped into the Bowman's capsule. We reabsorb it. So only 5% goes into the urine. Okay, so let's talk about what is going on first in the early proximal tubule and then the late proximal tubule. So let's talk about what all these different symbols mean. Okay, so the first little green blob, if I put ATP in it, what do you think it is? What's the most common ATP pump we have? Yep. Sodium potassium pump. This rectangular up and down one in red is a co-transporter. The oval one is facilitated diffusion. The purple oval is an aquaporin channel, aquaporin 1. What do you think they put through it? Water. And the brown pyramid one is a counter transporter or co-transporter. Oh, not counter, I'm sorry, antiporter. And the last one, the little simple, the equal sign, is just your simple diffusion. So that's just a key for all the symbols that are in the rest of the drawing. So let's talk about the early proximal tubule and its relation to getting all the substances back in. So we know the top one is an ATP. So which direction is sodium going to go? Out. And it's going to bring potassium in. As far as setting this up, this is the lumen of the tubes. Oops, sorry. So lumen, wall, this is the interstitial space. And then we have our blood. Remember, we're reabsorbing stuff, so it has to get back into the blood. So we start in the lumen, which is the tubular fluid, through the basement membrane or the wall. That's where remember that cell wall, that's where all the transporters are. Interstitial space, and then finally back into the blood. So the first one, ATP, we know sodium is going one direction, and potassium is coming in. The next co-transporter is going to be sodium and glucose. And glucose, I'll change the color to red. Glucose comes through, they both go through together, and then the glucose goes out by itself through facilitated diffusion. So sodium and glucose, co-transporters, go together, and they can get out. The next one down, sodium again. And your amino acids, they both go out, and then the amino acid goes out through the simple diffusion, or facilitated diffusion. Co-transporters, they go together. So you have to have salt in your diet. You need salt for all these functions. We get to the GI system, the way we absorb nutrients out of the intestine. Similar transporters. They take sodium and it goes along for the ride. Sodium is needed in our food for absorption of all of our nutrients. The last one is, again, sodium, but this time it's acids. Oops. And then those inorganic and organic acids go out that way too. Bicarb also, remember bicarb is also made by the kidneys, will go out by itself through facilitated diffusion. Here we have, the last one is a hydrogen with a sodium transport. So sodium goes out, hydrogen goes in. So we have a sodium hydrogen antiport, or counter transport, opposite directions. It's going to bring hydrogen into the urine. What's that going to do to the urine? Acidify it, which means we probably needed to get rid of some acidity in our blood. So we had to dump more hydrogen into the urine to get rid of some acidosis in the blood. The last two are your aquaporins. So we have water here. So water can go through by itself, and water can also get through the spaces in between the little fenestrations. Sometimes water can leak out in that way too. So that's how in the proximal convoluted tubule, sodium handling is working. So all that sodium that comes in to the wall is then pumped out with the gradient with the sodium-potassium ATPase pump, and that keeps all your gradients set so that sodium has a gradient to go into the wall. Remember, sodium is going along. There's no energy getting sodium and glucose in together. They just go. Because the gradient is set up, and with that sodium-potassium ATPase pump always working, it's going to get rid of that sodium that comes in, so sodium will always move into the wall. Again, that's why that sodium-potassium ATPase pump is important, because it keeps all of those concentration gradients stable. In the late proximal tubule, it's a little bit different. Don't have that much there. Still have that sodium-potassium ATPase pump. In this end, we have another sodium and hydrogen cotransporter. We have a chloride with a formate cotransporter. We'll learn about formate later when we get to the other titratable acids and so forth. Just know we have sodium and chloride going into the wall at this point in the late proximal tubule, and sodium and chloride by itself, they can also go through the fenestrations in between the walls as well. So that's how it works in the late proximal tubule. When we get to the thick ascending limb, now we're going to start talking about how diuretics work. So remember, there's different areas. The thick ascending limb, the proximal, and the distal convoluted tubules all have ways to reabsorb sodium. We went through already the proximal and the distal. Now let's talk about the thick ascending limb in the loop of Henle. This is a triple transporter, this first big one. It takes sodium, potassium, and 2 chlorides. They all occupy the receptor, and once they're all seated, they all go. All 3 ions have to be present for this transporter to work. Okay, so that's the first step. Then, again, we have our sodium-potassium ATPase pump keeping the gradients working. So, when we talk about loop diuretics, these are diuretics that affect the loop of Henle. All they do is when you give someone a loop diuretic, it blocks the chloride from binding. And when you block the chloride from binding, the whole transporter shuts down. So where's sodium going to stay? In the tubular fluid or go into the blood? Sodium is going to stay in that tubular fluid. If sodium is staying there, where's the water going to stay as well? In the tubular fluid, and that's how you diurese with a loop diuretic. It blocks the chloride from binding to the receptor, so the whole transporter shuts down. Sodium stays in that tubular fluid. If sodium is staying there, water stays there as well. We're not going to reabsorb that water too because sodium's not coming out. It's called diuresis. The water stays in the urine, and you just pee off the excess fluid. In the early distal tubule, we have a sodium, let me change that. Sodium and a chloride transporter. This is in the early distal tubule. Remember the loop of Henle was a triple transporter. In the early distal tubule, it's just a co-transporter with sodium and chloride going together. In this group class of drugs, they're called thiazide diuretics. So thiazide diuretics act at the early distal tubule. Example of that is hydrochlorothiazide, HCTZ. Okay, people like to use that in like med dramas because it sounds cool. They'll use it for everything. Oh, they're dying, they need a heart attack. Give them HCTZ because it sounds cool. So a thiazide diuretic acts at the early distal tubule. And again, same thing as before. It blocks the chloride binding site. Chloride can't bind, the transporter doesn't work. You need both of them there. Once you block the chloride, sodium is going to stay, chloride is going to stay in the tubular fluid, therefore water will stay there as well. And again, sodium potassium ATPase pump is always present again to keep those concentration gradients stable. The last one, late distal tubule and the collecting ducts towards the end. What influence plays the biggest role with reabsorption in these areas? Hormones. These are hormonal influenced. So when we get to the part, you'll see what happens there. So on this end, we have simple diffusion with potassium and sodium wanting to move. However, this is under the control of aldosterone. Okay? So aldosterone is a hormone that has the effect here. This class of drugs are called potassium sparing diuretics. When you give someone a potassium sparing diuretic, for example, spironolactone, it ends like aldosterone, spironolactone. It's an aldosterone type drug. It blocks this channel from occurring. So when you give spironolactone or a potassium sparing diuretic, it shuts that off. Where is the sodium going to stay when you shut that off? Where is the sodium going to stay, in the tube or back in the blood? In the filtrate or in the blood? Filtrate. Where is the potassium going to stay? In the wall, but before that, it has to come from the blood. It's this transport. So potassium, in this case, is going to stay in the blood. If potassium is staying in the blood, it's potassium sparing because in those patients, their potassium will stay elevated. Some patients you give diuretics to, their potassium levels drop. So if they have these sensitivities to different diuretics, you can give aldosterone, spironolactone, for example, and that will help give a diuretic effect, but it helps keep the potassium in the blood because we're shutting down this gradient here. The sodium moving out, potassium going in, we shut that all off, potassium stays in the blood, you lose sodium in the urine. And that's the effect of aldosterone. Okay, it's going to keep sodium in the blood. So you keep water there. If you keep sodium and water in the blood, what happens to your blood volume? Goes up. If your blood volume goes up, what happens to your pressure? It goes up. What is the cause for renin to be released? That starts the whole renin, angiotensin, aldosterone axis. What's the impetus? What's the stimulus for renin to be released? Low blood volume. Low blood volume going into the glomerulus is sensed by the macula densa in low blood volumes, and it releases renin to release aldosterone at the end to bring up your blood volume. Spironolactone is an antagonist to aldosterone. It blunts the effects of aldosterone. We don't want the pressure buildup. We don't want the volume in the blood. We want to get rid of it. So you give them that spironolactone. Sodium can't move. Therefore, water stays in the filtrate, and you just pee it out. And the excess water leaves the body, reducing edema. So that's in the late distal tubule how that one works. So looking at the drugs at a level of the entire nephron, you can see where these diuretics take an effect. Carbonic anhydrase inhibitors we haven't really talked about. What is carbonic anhydrase? What does it do? Remember, it's that enzyme that helps make CO2 plus H2O into the carbonic acid. And then it'll also dissociate carbonic acid into bicarb and a hydrogen ion. When you block carbonic anhydrase, it's really only used for like high altitude mountain sickness. When people go to like Everest and they start getting fluid on their lungs, they give them this type of drug, a carbonic anhydrase inhibitor, that will help diurese those patients. It's usually for that type of thing. Osmotic diuresis is just obviously what it sounds like, just doing with osmosis. But remember, these are the sites: sodium, 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 sodium. All sodium sites of reabsorption, and that's where we have the diuretic effect. And that's why those diuretics work in those areas. So thiazide diuretics, HCTZ is an example. Loop diuretic, this is your Lasix, your furosemide. That's a loop diuretic. It's going to affect the sodium, potassium chloride triple transporter in the ascending loop of Henle. And that's how diuretics work. So going back to that question I had yesterday, patient comes in, fluid overload. You give them a drug that's going to cause diuresis. How does it work? If we gave them furosemide, it's going to work where? Lasix, loop diuretic, thick ascending limb. If I say we have to give someone a potassium-sparing diuretic, they work at the collecting duct, late distal tubule. And again, these are just sites of sodium reabsorption. We can alter that with drugs to alter your fluid levels. Any questions about sodium handling? Okay. So here are just some examples of drugs. Again, your loops, furosemide, bumetanide, which is Bumex. Again, inhibition of that triple transporter in your thick ascending limbs. This is a good summary slide to study from. And then some examples of use and so forth. Here's your thiazides, potassium sparing, where they work. Again, aldosterone receptor antagonist. It's going to block the effect of aldosterone. And your carbonic anhydrase inhibitors. Okay. Okay. Let's do a little bit of water handling, and we'll go from there. So we have to balance the amount of water in our body. Obviously, we need water for all processes to occur. Allows for diffusion of things, and so on and so forth. 3 processes are required. Let me change the colors here. For this to occur. First, we need adequate GFR. We have to perfuse the kidneys. They have to have water, fluid, electrolytes going to the loop of Henle so we can see what our water stores are like. We also need the ability to have sodium reabsorption without water reabsorption in that ascending limb. This allows to dilute your urine by just taking in sodium by itself and leaving the water there. Allows you to get rid of fluid. If our medullary interstitial fluid becomes hypertonic, so what does that mean regarding salt? Is it too high or too little compared to reference? Too high. We have the potential to concentrate urine in the collecting ducts. Again, that's why it's under hormonal control, and that's why we're going to talk about the hormones right now. So water permeability must be variable in that common collecting duct, and it's controlled by ADH, antidiuretic hormone, also known as vasopressin. So if you ever heard of vasopressin, if you're an EMT before, vasopressin, ADH, they're the same exact thing. So we're going to be talking about ADH. So this is under the influence of plasma osmolarity. What's the concentration of solutes to the solvent floating around? We sense it in the hypothalamus. Hypothalamus sends the signal to the posterior pituitary to release ADH. We have large increases in ADH secretion if the osmolarity increases. If osmolarity is going up, what's happening to the amount of solutes in that solvent? It's increasing, it's going up. That means that either we added a bunch of solutes to the solution or we took water out. So if we took water out, we're losing water somewhere, so we release anti-diuretic hormone. We don't want to diurese. We don't want to lose fluid, so we want to retain it. Anti-diuretic hormone. It's going to make us keep up that water in our blood volume, not lose it to the urine. Therefore, ADH has its effects at the kidney. Allows for formation of concentrated urine and retention of solute-free water in the body. We're just absorbing free water. There's no salt in it, no potassium, nothing. It's just those aquaporin channels we talked about before. They're just water channels, and they're under the influence of ADH. ADH, and the reason why we use it, vasopressin in life-saving, which not really anymore, we don't use it as a first-line therapy anymore, but it's a potent vasoconstrictor. The V1 receptors promote vasoconstriction, but the V2 receptors, renal water retention. So let's talk about ADH again, vasopressin. Now we know its two roles, vasoconstriction and free water retention. If we're in a low blood volume situation, low volume, what do our vessels have to do to maintain that pressure when you have a low volume? They have to constrict to keep your pressure up. So ADH, by causing vasoconstriction, helps keep your MAP up by causing that vasoconstriction. The second job of ADH is going to go to the kidneys and say, hey, we have low blood volume. We need more. Stop peeing water. And it's going to have its effect on the kidney to bring water back into the blood, bringing blood volume up. Therefore, the pressure comes up as well and all that fun stuff. So that's the role of vasopressin. Now, how does it come out? What are the causes? So, synthesized in the hypothalamus, nerve terminals in the posterior pituitary. There's two sides to the pituitary gland, anterior, posterior. There's only two posterior hormones, but they're both with neural mediated, meaning a neural response will get those hormones secreted fast. What's the other one from the posterior pituitary? Oxytocin. Oxytocin. Those are neurally mediated. They're fast acting. All the hormones on the anterior side are hormonally mediated. You need stuff to go through the blood to get to that pituitary gland, activate those receptors to have a response. Anterior pituitary is very slow. So whenever you see anything saying it's neurally mediated, it's going to be a fast response. Reduction in plasma volume of more than 10% is a stimulus. We'll figure that out via our low pressure receptors in the atria and the lungs, and our high pressure receptors in the carotid baroreceptors. And that's how we figure out what the pressure is like in the system. Remember, we have pressure systems, pressure things. Nicotine causes increased secretion of ADH. So what does that mean to people who smoke all the time? Are they going to pee a lot or less? Less. What's ethanol? Alcohol. What's breaking the seal? Why do you think you break the seal? It's ADH. ADH inhibits secretion of ethanol, inhibits secretion of ADH. So when you're drinking, ethanol tells the brain, we don't hold back on our ADH hormone, so we lose it all in the urine, and then you start peeing and you keep going. It's all ADH that does it. So there's a couple of issues that can happen with ADH hormone. One is called syndrome of inappropriate antidiuretic hormone, SIADH. They just keep making this hormone. So retention of solute-free water in the blood, eventually get hyponatremia, so on and so forth. You can have problems with ADH secretion. We'll get to this more when we get to endo. So how does ADH work in concentrating or diluting our urine? Remember I said yesterday, our body doesn't care about the urine. It's a waste product. We're getting rid of it. So concentrating or diluting the urine has no effect on us per se. The main goal is regulating the extracellular fluid compartment. We can get rid of excess things via the urine. So if we increase plasma osmolarity, leads to urine that is more concentrated than plasma. Why do we have a concentrated urine? What are we doing to the volume of that urine? What are we doing with the water? We're pulling it out because it's got to go in the body because our body says we don't have enough water. So we're going to make concentrated urine, pull out the water out of the urine, keep it in the body where we need it because we need to get rid of that, lower that osmolarity. So some solute-free water returns to the ECF, and plasma osmolarity reduces. So concentrated urine, eventually it's going to reduce osmolarity because that water goes back into the blood and increases the solvent of that osmolarity side, and we go from there. Opposite's true. If we have a decrease in plasma osmolarity, now we have what? Too much solvent or too much solute? Too much solvent, too much water. It's diluting out our blood. We don't need all that water. We have to get rid of it now. So we excrete dilute urine. We're going to get rid of the water. It's going to pee it out. It's going to dilute out the urine. Therefore, some solute-free water is excreted in the urine, and the plasma osmolarity is going to increase because we're getting rid of the water in the blood, and we're just peeing it out. Whether the kidneys produce concentrated or dilute urine depends on the actions of ADH. It's all under that influence. So in stages of water deprivation, and after this we'll stop and take a break, and then we'll do the Kahoot. Okay, so water deprivation. In this case, let me change the color to blue. We have a low amount of water in the body. Okay, this whatever percentage this is of the body is water. In this case, what have we done to the solutes? Have they gone up or down? If we have low water in the body, what happens to the solute concentration? Goes up. So in this case, we have an increased osmolarity. Okay? This increased osmolarity goes to the hypothalamus. And then obviously later on, that's your pituitary gland. Two things are going to happen. What are two things our body does when we need water? One we just learned about, ADH. What else do we do? What happens to us when you get dehydrated? Thirsty. So one response, increased thirst. You drink water. That's one of the things our body's going to do, increase our thirst. So whenever you're thirsty, drink water, otherwise you're fine. When we put too much emphasis on water, we're fine. Just a way to sell bottles. The other job, obviously, is going to be ADH comes out. ADH has its effect, remember, at the very end, common collecting duct. What's going to happen to water? What are we going to do with the water here? Are we going to reabsorb it or excrete it? Reabsorb it. Increase reabsorption. So what's the urine going to be like with regards to volume? Low volume of urine. But what about the osmolarity of this urine? High or low? High. This is a concentrated urine because we need that volume, that liquid, that water back in the blood. Let me put this up. So this is what happens with water deprivation. Let's talk about water excess now. We have too much water. Okay, now we're filled up. We have fluid overload. We have too much water in the system. What happens to our solutes when we have too much water in the system? What happens to the concentration of these solutes? When we have too much water, decrease. So we decrease solute concentration. Therefore, what happens to osmolarity? Decreases as well. We're adding a lot more water to the system. It's going to dilute it out. Osmolarity goes down. So why is it important that we say solute concentration and not the number of solutes? What is changing? The water. The water, the volume. So sometimes in fluid overload, it can even show you that you're anemic because you're adding so much water to your blood volume. It can dilute out your red blood cells too and give you a false reading that you're anemic. Once you replace the volume, then the anemia goes away. Because you're decreasing the concentration because you're adding too much water. So sometimes you get false readings. You have to look at your volume status of the patient. Are they euhydrated? If they're really dehydrated, it alters their bloods. So with a decreased osmolarity, what is this going to do to the hypothalamus? Is it going to stimulate it or inhibit it? With a low osmolarity, do we want ADH to come out? No. So it's going to block ADH, and it blocks the thirst. We don't get thirsty because we already have too much water in the system. We don't need to drink any more. And obviously with that low osmolarity, we're not going to have ADH come out because we don't want to retain more water. We have to get rid of water. We have to diurese ourselves. So we don't want ADH to come out. ADH doesn't come out. What happens in this case? We have our urine come out. What happens to the volume of the urine now? It's going to go up. We're going to pee a lot more. What happens to the osmolarity of the urine? This is of the urine osmolarity. The goal is to raise the osmolarity in the blood by getting rid of the excess water. We're getting rid of the solvent. We're getting rid of the water. So we're getting rid of it in the urine. Urine volume goes up. That's why you start peeing when you break the seal when you're drunk. And it's clear because it's very, very dilute. There's not a lot of solutes in it because you're retaining everything. Okay, so that's water balance. Here's a case called diabetes insipidus. Basically, this is the opposite of the syndrome of inappropriate ADH. Now we don't have enough. Don't worry about this stuff so much, but know that there's different types of diabetes insipidus. But look at this. Patients with DI may excrete 20 to 25 liters of water a day. They are peeing all day when they have this problem. Okay? And don't worry about that. Okay, we're going to, let's see how long. Yeah, we'll stop here. Pick up tomorrow. So come back at 1:20, and we'll do the Kahoot.